Hello everyone, welcome to the BI459 podcast, where today's hormone discussion will be centered around somatostatin. Somatostatin is an integral hormone in the body as an inhibiting agent key to maintaining homeostasis in the body. Somatostatin has been documented to be produced in the pituitary, pancreas, GI tract, hypothalamus, and central nervous system, applying its inhibiting effects in areas such as the GI tract, endocrine, exocrine, pancreatic, pituitary, and central nervous system secretions.
I would like to mention that prior to researching, our group did not have much insight on how somatostatin played a role in the body, as it's not a common hormone known to the general public. I feel like a lot of these inhibiting hormones really go unnoticed, but I'm glad we're bringing some light to them.
I feel the same way, Andrew. Today, I have actually brought in some research experts and even two special guests to cover all of the ways it works in our bodies. To start off, Maddie, one of our research experts, will be discussing some of the important features and basic functions it has in our bodies.
Hi everyone, I'm Maddie. So somatostatin is a cyclic peptide which possesses neuroendocrine inhibitory effects. There are actually two different active forms varying in their amino acid length. The shorter isoform acts primarily on the brain, while the longer isoform acts primarily on the gastrointestinal tract, where they both have similar inhibitory effects and activity. It can also be used clinically in the treatment of neuroendocrine tumors when more stable isoforms with longer half-lives are produced.
Wow, I didn't know that it had such a huge impact on the human body. Could you enlighten me and the rest of the listeners today on more of the basic functions, please?
Somatostatin is generated in multiple different locations, which many people don't know, including the central nervous system, the gastrointestinal tract, pancreas, and surprisingly even the hypothalamus. So, in the central nervous system, somatostatin has the ability to alter memory formation and even neurotransmission. It has anti-proliferative effects on both healthy and cancerous cells in humans and animals, and even works to prevent angiogenesis, which is the formation and growth of new blood vessels.
Okay, that's very interesting to learn. Thank you for sharing. I did do a little bit of research on my own before inviting everyone today and found some interesting points. I found out that somatostatin plays an essential role in the regulation of the endocrine and exocrine systems by inhibiting the secretion of specific gastrointestinal, pancreatic, and pituitary hormones, which inhibits their activity, which you did touch on before. I know you also said that somatostatin is found in the hypothalamus, but fun fact for everyone today is that it was actually the first place somatostatin was identified. The anterior pituitary, that is also located in the hypothalamus, is responsible for the inhibition against growth and thyroid-stimulating hormones as well.
Oh wow, I didn't know that. I thought you were just the host today. Since you've done your research clearly, can you briefly discuss how it affects the parasympathetic and sympathetic nervous systems?
Oh yeah, I actually have some information on that too. Somatostatin is released by the two neural mechanisms, parasympathetic and sympathetic nervous systems, but it has opposing effects on them. It actually uses the sympathetic efferent nerves to inhibit pancreatic function. For the parasympathetic system, it uses cholerogenic agents to directly inhibit the function of B cells, which I'm sure we'll talk about later. It also affects the secretion of parental cells as well.
That's really interesting. Since you mentioned it was also found in the pancreas, I want to touch more on its role and what makes it so important in the pancreas. The reason it is so important is because the Langerhans are found within the pancreas in specialized compartments. Insulin can also play a part in inhibiting the secretion of somatostatin and glycogen by binding to the B cells in the pancreas, because insulin receptors are coupled to tyrosine kinase. Insulin then functions in the process by affecting glucagon secretion and regulating blood glucose levels. Since both insulin and glycogen reversibly affect each other, when there is a decline in blood glucose, glucagon is secreted, and when there is a rise in blood glucose, insulin is secreted. Since the B cells in the pancreas are excitable cells and have a depolarizing factor, they have the primary role of insulin secretion. Also, alpha-beta cells, which are responsible for the production of somatostatin and inhibition of glycogen and insulin by their adjacent cells.
Wow, and to think I was just the host on a podcast today. I have learned so much already, and I'm now going to introduce you guys to our very first special guest, Louisa.
Of action of somatostatin, as this is my specialty. So let's just get straight into it. You guys have already spoken to the fact that somatostatin has the ability to inhibit cell secretion and proliferation, as well as induce apoptosis. But the reason these things occur to maintain homeostasis is due to their receptors. So there are 5 receptors, and their abbreviation is STTR, but for simplicity's sake, I will just be naming them 1 through 5. All of these receptors are G protein coupled transmembrane receptors. So what that means is these receptors go all the way from the extracellular fluid into the intracellular fluid, but are in the membrane on the outside of the cell. All of these are found throughout all of the human body. However, receptors 2 to 5 are the most predominant in the endocrine tissues. I'm going to go into what each of these receptors is responsible for because they are all different. The first receptor triggers a cascade which inhibits the release of growth hormone, prolactin, and calcitonin. The second receptor inhibits the secretion of growth hormone, which is similar to receptor 1, as well as the inhibition of adrenocorticotropin, glucagon, insulin, interferon gamma, gastric acid. So, similar to the first two, the fifth receptor also inhibits growth hormone, adrenocorticotropin, insulin, but it also has the ability to inhibit amylase and glucagon-like peptide 1. In contrast to the first 3 receptors I mentioned, the third receptor reduces cell proliferation and helps induce apoptosis in appropriate cells. Finally, the mechanism involving the fourth receptor is still not very widely known and still requires further studies, so maybe on a future episode we could discuss the fourth receptor. So if you take a look at the diagram I have made here, in general, you will see that there are 3 potential outcomes when somatostatin binds to one of its receptors. There could be a decrease in cyclic AMP, a decrease in calcium influx, or the stimulation of phosphatases such as calcineurin. Ultimately, all of these pathways lead to an inhibition of exocytosis or they induce apoptosis. So, the reason cyclic AMP decreases, causing an inhibition of secretion, is because adenylyl cyclase is inhibited first, and then in the event that we are looking at the ion channels, when the calcium and potassium channels open, there is a decrease in calcium influx, and this leads to the same outcome but with a different pathway. So both the adenylyl cyclase and the ion channels being open lead to proximal effects on the decreasing of cell secretion via exocytosis. When the activation of a receptor uses the phosphatase pathway, there's an increase in serine and threonine phosphatases as well as calcineurin. With an increase in calcineurin, you will see a distal but direct inhibition of exocytosis. So at the end of the day, all 3 of these pathways lead to the same outcome, but all of these are based off of what the body needs to maintain their homeostasis. So I hope this little schematic and information helps you out with what you need to know, and hopefully we can meet in another episode and discuss receptor 4 one day.
Thanks, Louisa. Now we'll hear from our other special guest, Abisha.
Hi, I'm Abisha, and thank you so much for inviting me on the podcast today. Where do I begin? Discussing the metabolic effects of somatostatin is such a unique and cool topic when it comes to our body's endocrinology. For starters, somatostatin is created by D cells within the pancreatic islets, which apply a neighboring pancrean inhibitory impact on hormone discharge by the local glucagon secreting and B insulin secreting cells, hence planning carbohydrate metabolism. The major component by which somatostatin represses afferent emission is to diminish calcium convergence through a cough-like toxin-sensitive hyperpolarization of B-cell potential. In addition, somatostatin hinders the discharge of afferent glucagon and pancreatic polypeptide from endocrine pancreatic cells. So we see somatostatin controls the discharge of other hormones that are included in the digestive system, such as thyroid-stimulating hormone and cortisol. Generally, somatostatin makes a difference to direct the change of hormones that are included in the digestive system, which makes a difference to preserve large metabolic homeostasis. It is then broken down by chemicals called peptidases, which cleave the peptide bonds within the hormone to make small peptides and amino acids once somatostatin has been broken down. It can then be disposed of by our body through our kidneys, which is a pretty amazing fact. After somatostatin is discharged into the circulatory system by the pancreas, it then voyages down to the liver where it is metabolized by peptidases. Inactivation is also really important as is it a hormone that regulates the release of other hormones and occurs primarily in the liver as well through the process of degradation by enzymes. Proteins may play a vital percentage within the control of polypeptide hormone levels, and the peptidase acting on these 4 hypothalamic hormones may direct the sum of thyroid releasing hormone, luteinizing hormone releasing hormone, and release inhibiting hormone. And somatostatin is discharged from the hypothalamus or their activity at the level of pituitary and their evacuation from the circulation. By examining the peptidase proteins, advanced data may be taken on the physiological instruments controlling the emission and activities of hypothalamic hormones, as well as the plan of analogs with expanded or competitive activity. So to better understand all this information, inactivation of somatostatin research study on brains of mouse were studied, with the accessibility of a sensitive and radioimmunoassay for developmental hormone release inhibiting hormones. And these are conceivable to explore the nearness of peptidase proteins competent of inactivating the hypothalamus and other brain zones of the rodent. It was found that both the rodent and the particulate divisions from the male rodent hypothalamic rapidly inactivated somatostatin, which the chemicals included have an ideal pH of 7.3. Other than the affirming the nearness of peptidases inactivating the release inhibiting hormone within the hypothalamus, the image that is shown explains the results may demonstrate that somatostatin encompasses a significant exterior from the hypothalamus anterior pituitary axis, but inside the central nervous system. So I hope this helps expand your knowledge on the hormone and gives you a new and improved insight on the metabolic process. Thank you so much for having me.
Thank you. Somatostatin, as mentioned now, plays a pivotal role in inhibiting hormone releases from most neuroendocrine cells. If there were to be a chronic decrease or increase in this inhibiting hormone, one can assume that there would be countless body-wide problems that would arise, right? What would be your guesses?
Well, somatostatin is in charge of inhibiting many other secretions, so if it's hyperactive, I guess I would assume that there would be a lot less important hormones in the body.
That's exactly right. This chronic hyperactivity of somatostatin has been noted in medical journals as a byproduct of the cancerous formation of functional somatostatinomas. Historically, the first case was recorded in 1977 in a 46-year-old woman in the head of her pancreas, which started the beginning of the medical literature on this cancer. At the time, the doctors, having never seen this before, were able to find this tumor through a computerized tomography scan of her pancreas. The picture shown here shows a transversal widening of the pancreas, indicating the doctors of a tumor. I also have a comparison picture here to show the difference between a normal pancreatic CT scan and the injured pancreas.
Wow, those pictures are incredible.
Since then, the vast amount of somatostatinomas have been recorded to originate in the head of the pancreas and a minority in the body and tail, as well as a few cases in the duodenum, ampulla or vatar, the small bowel, with extremely rare instances in the extragastrointestinal primaries in the lungs, kidneys, and thyroid.
So what causes these cancers, and who exactly is most at risk?
For some background, this cancer has a rare occurrence with an incidence rate of 1 in 40 million and accounts for less than 5% of pancreatic cancerous tumors, and has a common emergence in late adulthood with no gender sway. The etiology or cause of this cancer has been associated with the neurocutaneous syndromes. Johns Hopkins Medicine characterizes these as congenital syndromes.
Sorry to interrupt, but does that mean it's present at birth?
Yes, it does. So these congenital syndromes affect the central nervous system, bones, muscles, and organs, and cause a variety of adverse symptoms and increase the probability of developing tumors. Of the various neurocutaneous syndromes, the most common association is with multiple endocrine neoplasia type 1, followed by neurofibromatosis type 1, and von Hippel-Lindau syndrome. In addition, these tumors can be sporadically conceived and is associated with the loss of heterozygosity at chromosome 11q, 6q, and the allelic loss at chromosome 3p. Moving to the characterization, somatostatinomas are divided into two subsets of functional and non-functional. Non-functional tumors are identified as tumors physically obstructing other organs and damaging the homeostasis in the body, but do not involve the secretion of any biological hormone. Focusing on the pancreas, functional tumors, on the other hand, develop in the D cells of the Isle of Langerhans of the pancreas. multiplying rapidly and releasing a large amount of somatostatin into the body. The result of this abundance of somatostatin in the body leads to the clinical manifestation of cholelithiasis, diabetes mellitus, weight loss, and steatorrhea and gallstones.
That sounds horrible. What treatments do we have for this?
I must preface, I'm not a medical doctor or a pathologist, so I can only say that there are treatments available and you should consult a doctor if you're faced with any of the symptoms. And it also should be mentioned that synthetic analogs have been created to reduce the symptoms of other types of cancers, such as glucanoma, insulinoma, growth hormone releasing factor tumor, and many others. These analogs play an important role in treatment for these tumors to regulate the production hormones these functional tumors produce.
That's very interesting. Now, you've mentioned the problems with an abundance of somatostatin in the body, but what if there's deficiencies?
So very little cases have been reported about somatostatin deficiencies, but the few occurrences I've researched mention in Alzheimer's, somatostatin has been seen as a role of a neurotransmitter in the brain, and Alzheimer's has been seen as a cause in the decrease of this neuropeptide, although the mechanisms of how this functions is unclear. Apart from that, I haven't seen anything in medical journals about somatostatin deficiency. From my non-medical undergraduate brain, if there were to be a deficiency of somatostatin, there should be signs of hyperactivity in all the hormones somatostatin suppresses.
Well, thanks, Andrew, for shedding some light on somatostatinomas.
Before I go, I want to show a picture of the tumors on the pancreas for some potential future doctors or anyone else who enjoys the sight of tumors.
So there's a lot of information that we've been talking about here today, so I wanted to highlight some of the key points to summarize. So we know out of the two isoforms that the shorter one acts primarily on the brain, while the longer one acts primarily on the GI tract, but they do have similar inhibitory effects in both locations. Along with the functions of somatostatin, analogs can be used for treating neuroendocrine tumors. So somatostatin is generated in multiple locations, as we mentioned, such as the central nervous system, the GI tract, the hypothalamus, and the pancreas. Glycogen acts as a positive effector for somatostatin and insulin secretion, but somatostatin exerts a negative effect on insulin and glucagon secretion. We've talked about how insulin, glucagon, and somatostatin work together to regulate secretion in the body and maintain our homeostasis. As I was speaking to earlier, there are 5 different receptors that somatostatin can bind to, and depending on which one is activated, it triggers a different cascade to achieve either an inhibition of cell secretion, proliferation, or the induction of apoptosis. The 3 possible pathways include the adenylyl cyclase cascade, the ion channel cascade, and the phosphatase pathway. As the other expert with us here today had mentioned, somatostatin is a hormone made by cells in the pancreas that helps control the quantity of other hormones released in the body. This includes hormones involved in breaking down and using carbohydrates and maintaining stable metabolic levels. This happens by lowering the release of hormones like glucagon, insulin, and thyroid-stimulating hormone by lowering the calcium levels in the body. Somatostatin is broken down by peptidases and deactivated in our liver. The activity of these peptidases affects the amount of hormones in the bloodstream, therefore regulating our homeostasis. Given that somatostatin has inhibitory effects on other hormone secretion, a chronic increase or decrease in levels would result in some sort of disruption in homeostasis. So, chronic increases in somatostatin have been attributed to the formation of functional somatostatinomas. These tumors can be functional, which means that they release large amounts of somatostatin, or they can be non-functional, which means they physically obstruct the other organs, but they don't secrete hormones. There aren't many documented cases of somatostatin deficiencies, but there has been mention of their involvement in Alzheimer's. On a positive note, somatostatin analogs can be used for the treatment to inhibit the growth of other types of cancers. I know that's a lot, but just a quick summary for you guys, make it a little easier to put all that information together. But it was a pleasure joining you on this podcast this afternoon, and I hope we all learned something new today.
Well, that's the podcast, folks. Thanks for everyone for joining me today, and a special thank you to our guest speakers and our co-hosts. We would also like to thank our sponsor, Dr. Georgiou from Wilfrid Laurier University, for all of her contributions to make this podcast today. Thanks, everyone.